Thank you, Richard, for having put together this interesting meeting and also for having me part of the conversation. So I'm going to discuss with you this idea that we call environmental Galenics. And if you've never heard of it, you will learn in a couple of minutes. So I come from Madrid. I work in the National Center for Biotechnology in Madrid. And for a long time, our laboratory and a bunch of other laboratories in Europe have been interested in developing bacteria in the laboratory as agents for bioremediation. And this idea and this concept comes from a long time ago. And the result of all these investigations is that we are able to produce in the laboratory at the level of a Petri dish very nice bacteria able to degrade a whole bunch of interesting pollutants. But the big problem is how to scale that up at a level that makes a difference in terms of bioremediation. So these bacteria or these microbes that we can train in the laboratory can be used for, in principle, have the potential to work in different scenarios. And thanks to the technologies of synthetic biology, as you know, now many problems that before were untractable, now there's a perspective that perhaps we can tackle them through these advanced agents for remediation. It includes CO2 capture, plastics, water, nitrogen capture, you name it. And in this area, in California and in the Mediterranean area also, there's this growing interest to do something with scorched soil after fires. So, but there is this issue on how we pass from going at the level of a Petri dish or a small bioreactor all the way to a wide area. And this is the idea of environmental Galenics, that is about how to formulate and spread symbio agents, or at least agents that we have prepared, trained in the laboratory for this type of applications. So what is Galenics? So Galenics is a Galenic science that comes from pharmacy, and is about the challenge of how to deliver active principles to a sick body. So you may have a molecule that has something interesting to combat a disease, but you have to think on how you deliver that compound to the ultimate target. You want to have it in a pill, you want to have an injection, you want to have an aerosol, you can have a suppository, and this in itself is a major technological challenge. So under the same frame, the idea is how to formulate these new activities that we can develop in the laboratory in a fashion, in a physical format that are efficient in their activity in the environment, and eventually how we spread them at a very large scale. So we can think on having monoclonal agents. Obviously, for us scientists, is the ideal situation. But then we can think on engineering consortia. And as you know, with the tools of synthetic biology, this is becoming more and more, say, amenable. Or you can think in even developing whole systems where you have an ingredient that is a plant. You may have fungus, you may have bacteria, and you can even engineer different interactions among them. And this is something that would constitute a bioremediation unit. Or you can think even on DNA transfer. I will return to that in a minute. But this idea of how you deliver things into the environment at a large scale is what the topic of environmental Galenics is about. And in that context, there are various what you may call technical challenges. One of them, that it would be the low-hanging fruit, would be to develop monoclonal catalysts, where we engineer new pathways for new compounds. But you can also think on engineer consortia, not only bacteria to bacteria consortia, but also consortia bacteria to yeasts or bacteria to plants. And you can also think on combining the biological activities of microorganisms with the catalytic activities of other abiotic catalysts. And the possibility is there, and there are different technological approaches to do that you can develop these mixed biocatalytic materials that have a potential for the environment. But then how you spray that at a very large level? So obviously the kind of default procedure is to make a solution of your favorite agent, and then to spray that with the various technologies that have been developed out there for spreading things in fields or in agronomical context and so on. But you can think in many other possibilities. If you want to spread something interesting from a biotechnological and environmental point of view in a large extension, you can think not only on coating seeds, for instance, for having plants that are colonized by bacteria that do something to environmental pollutants. You can even think on using insects as agents for propagation. You can even think on engineering clouds, and you may know that there's quite a few literature on how bacteria can be spread out in large extensions through clouds. And you can think also on sand, storms, and transportation of particles in the atmosphere. So you can think big, not on how to contain bacteria, but just the contrary, how to spread bacteria at a larger scale, provided that what they spread is something beneficial, not something detrimental. Obviously, the question that immediately appears is containment and biohazard and all these concerns about risk assessment. And, well, we have been discussing before, and this is a topic that pops up all the time, on risk assessment, containment. Okay, so I don't know how it may work in other biological systems, but in bacteria, we claim, and other people will support what I say, that there's no certainty of containment that a technology can deliver, at least in sight. So that means that in the microbial world, life will always find a way, regardless of how smart we are in having firewalls, in having alternative genetic codes, in having all types of circuits. Eventually, some of them will escape our barriers. And therefore, containment, in the opinion of a growing community, is not an ultimate choice for this type of things. So what can we think instead of that? Okay, so the proposition that is becoming more and more considered is that instead of putting the emphasis in containment, maybe we have to put the emphasis in tracing and being able to track the fate of the different agents that we put into the environment to a given origin. So by the same token that we put chips in pets, then maybe the idea is that we can barcode bacteria and microbes that go into the environment. It can be barcodes, it can be some type of water marking, some type of marks that we put in the genomes of the bacteria that are for environmental release, so that we can not so much contain them, but to trace them. So the emphasis goes from containment to traceability, and that's something that perhaps is more realistic. So there are many approaches to do that, or not many, but a few approaches to do that. And the approach that people like Natalia Krasnogor, with whom we have been collaborating recently, have proposed is to develop a computational, concept in which we can identify sequences that are entirely non-biological and they have not been associated at all to any biological, sequence, and then to use them as barcodes, as unique identifiers to putting in permissive sites of the genomes of bacteria or microbes to be released into the environment, and then use that as a kind of a marker to identify, if necessary, the species, the strain, the intellectual property status, the pedigree, and also, if necessary, to have a set of countermeasures in such a way that the barcode can take us to a kind of digital twin that immediately will guide our subsequent interventions to control the situation if something may go wrong. But the bottom line of this story is that certainty of containment may not work, and certainly is likely not to ever work in bacteria for environmental release, so we have to think differently. But we can think in releasing not only bacteria, but other things. And again, one of the take-home lessons in the earlier studies on implantation of recombinant bacteria into pre-existing communities is that existing microbiomes have this phenomenon that is called resistance to colonization. So that means that you have an ecosystem with bacteria that have reached a level of balance, then it's very difficult for newcomers to become established in this particular niche. Therefore, what if instead of trying to introduce a newcomer into a pre-existing ecosystem, what we try to do is to enrich, to fortify the existing microbiome with new activities that could be beneficial for the environment as well. And we know we can do that because we know that DNA propagates very quickly. And there are already many channels for horizontal DNA transfer that, under some perspective, are bad because they can transmit bad things, but they are there, and perhaps we can leverage them to transmit and propagate things that are intrinsically good, such as, for instance, activities for biodegradation of intrinsically toxic compounds. And on that basis, we and others have developed this idea that perhaps instead of thinking on how to stop horizontal gene transfer, what we can think is on how to domesticate horizontal gene transfer to fortify existing environmental microbiomes with beneficial traits through horizontal gene transfer mechanisms that, we like it or not, are there and will remain there. In that context, for instance, one of the projects that we have running in our laboratory is the development of some super promiscuous plasmids that can propagate recombinant genes in diverse soil microbial communities. And this is just one example. Obviously, I will not go into the details, in which we put a marker, GFP, in one of these plasmids, in one of these promiscuous plasmids, and then we put it in a plant pot, a small microcosm, and then you see that over time, this plasmid is propagated to many members of the existing community there. And what is more interesting is that you follow the kinetics of transfer of these plasmids and these markers. You observe that the initial inoculum that you use disappears more or less quickly, but then the genes of interest get transmitted over and over and over to different members of the community. So we think that this is a kind of indication that perhaps one of the lines of research and technological developments in this field is to domesticate and play with horizontal gene transfer as a way of fortifying the existing biodegradation abilities of existing communities, not so much to enter new members in a community. Well, this is something that you can do at a small level, but what happens if you want to scale this up at a very high level? Well, in reality, one of the technological, say, limitations of this approach is that we don't have good models of how we can really leverage this propagation of bacteria and, and propagation of genes at a high scale. And in that context, we have a collaboration with Ricard Soler of the University of Barcelona to find to make a model of what would be the right scenario, the right parameters that would enable a sound implantation of either new comers into an existing microbial community or propagation of beneficial traits through horizontal gene transfer. And in that context, again, I cannot go through the details, we have various models that will tell us that indeed that might work very well. And if you set a number of parameters in terms of survival, in terms of competition for resources with an existing community, at the end there is a situation where the newcomers may do well in terms of increasing the biodegradative activity and the biodegradative, say, status of a pre-existing community. Well, these are some details. If you're interested, we have some papers there. Okay, but we have to go on, and I'm happy that the previous speaker talked about the situation with the gene drives, because in fact, it's a bit naive to think that we can go straight from the level of having a culture in the laboratory and go straight into some type of release. By the same token that in the clinical and medical work, you have all these trials, all these clinical trials with different phases, phase 1, phase 2, phase 3, phase 4. Then we argue that to make release of bacteria for bioremediation or reality, we have to establish some type of legal frame, some type of conceptual frame in which we can take an activity that is created in the laboratory and to take that through various stages of tests, perhaps going first from at the level of a single Petri dish, and then move to a microcosm, and then to go to an ecotron, for instance, that are these environmental simulation systems that are out there where you can control many, many different parameters. And eventually, on the basis of phase 3, you can think on scaling up at a very large scale. And in the meantime, you have generated data that can be the basis for having some criterion for approval or not in release. So I think that all this could be organized properly and make a release of genetically engineered bacteria for bioremediation a reality. Okay, but then the final question, or one of the final questions is, well, you can have many, many patients, and you can try a drug in many patients, but how you can try a new synthetically produced agent or a genetically engineered agent in many environments, because you have such a large number of environments that it will be difficult to have data enough to train machine learning or artificial intelligence platforms to enable us to make sound decisions. Okay, so in that context, what we have proposed is to go a few steps back and perhaps to think a little bit on how we can define what is an environment in more rigorous terms. And if you go to the dictionary, at the end of the day, an environment is a scenario where you have a large but limited number of physical, chemical, and other factors that define in very precise terms what is going on in a so-called niche. So we wonder whether on that basis we could think on having a concept of environment, having a model of what is an environment by digitalizing or by pixelizing an environment in small portions where you can define very precisely what are the parameters that are at play in that particular location. So obviously you can go to different degrees of definition, but the idea is that many of these parameters can then be reproduced in a machine, for instance, in a millifluidic system where you have a tube, and in every tube you can simulate a number of parameters that determine the scenario, the physical, chemical scenario of a given environment, and then use these small droplets as the medium where you test the performance of a given environmentally interesting agent. In that way, you can see the performance of that particular agent, being one strain or a community or other agents, in thousands and hundreds of thousands of different conditions, and then you can generate enough data to feed a machine learning system that will guide, in principle, will have a predictive, say, ability to tell you what will happen if you now take this particular agent into a real scenario. Obviously, this is not the end of the story, but this is more than nothing and much more than we have at the moment, where we just basically rely on large-scale releases or microcons that you can handle only in a very small number. So to that end, we are putting together this what we call a high-throughput environmental simulator, where you can monitor the performance of a given agent for bioremediation in a large number of physical-chemical conditions by playing with these droplet-based kind of small scenarios, and where you have as an output a fluorescent output that you engineer with a fluorescent marker. Well, there are technical details and so on, but the idea is that if we want to have a predictive value of what will happen if we put something to environment, whether as single, as monoclonal agents, or as a consortia, or as horizontal gene transfer, we need to have a lot of data that is able to, or that are able to give us a criterion later of what would be the performance of a release, even beforehand the release itself. Okay, well, there are many technical issues connected to that. In fact, one of the claims that we have is that there's a lot of science that still need to be tackled to make these releases a reality. It's not just a regulatory barrier. We still have many different scientific questions and technical questions that we have to tackle and solve before we can make this idea of releasing things for environment a reality. And one of the approaches that we're trying to tackle is to see whether we can combine the data that comes from these experimental high-throughput simulations that I was telling you about with already existing data on the behavior of bacteria that inhabit in niches with environmental pollution. And we think that perhaps we can use these language models that are already available to convert descriptive data on the fate of bacteria that inhabit these locations to make it compatible with data that comes from these other experimental platforms. And at the end, our kind of ambition would be to use these artificial intelligence and machine learning platforms to produce digital twins that will guide the deployment and optimization of interventions on the basis of site-specific parameters. So I'm sorry I had to rush to this. Obviously, this is not a finished story. This is to be continued. And keep calm and thanks for your attention.